So let's talk in brief the synthesis, storage and final secretion of thyroglobulins, that is nothing but the thyroid hormones. So this is the blood vessel which is present just below the basement membrane. So this is the basement membrane of a one thyrocyte cell. This is a one thyrocyte cell. This is the adjacent thyrocyte cell, and these thyrocyte cells are arranged in a circular fashion, and in between these cells this is nothing but the lumen of thyroid follicle. So what is the first step in the formation of thyroglobulins? That is nothing but this thyrocyte cell. This is nothing but the thyrocyte cell. This thyrocyte cell collects a raw material from the blood vessel or from the circulation. So this thyrocyte cell collects a raw material from the circulation. This is nothing but circulation or blood stream. So the raw material comes from the circulation towards the thyrocyte cell. The raw material, for example, the raw material is nothing but the iodide. So the major source of iodide is nothing but present in the gastrointestinal tract or in a stomach. So from GIT, the iodide comes inside the circulation. So iodide comes into the circulation from GIT. So once the iodide enters into the bloodstream, this iodide has to be taken up by the thyrocyte. So this iodide is further go into the thyrocyte cell. This is not a simple process. That iodide is a charged molecule, that it has a negative charge. This iodide is a charged molecule, so this iodide cannot go directly or cannot diffuse or dissolve directly into the lipid. So there is presence of something which take up this iodide into the thyrocyte cell. About the whole iodide, about 20% of iodide is taken up by the thyrocyte cell, and remaining 80% of iodide is go into the urine. So from the whole iodide, 20% of iodide is taken up by the thyrocyte cell and 80% of iodide is excreted in the urine. So this 20% of iodide is cannot directly diffuse or directly dissolve into this lipid or directly go into the thyrocyte cell. There is a presence of some special channels, or there is a presence of some special mechanism before that. So there is a presence of, this is nothing but the pump. This pump is known as iodide pump. This pump is known as an iodide pump. These iodide pumps are present on the basement membrane of the thyrocyte cell. So this is one iodide pump. This is next iodide pump. So these iodide pumps are present on the basement membrane of the thyrocyte cell. So iodide molecule comes and finally enters into the thyrocyte cell. This is the next iodide. So finally, with the help of this iodide pump, iodide molecule, about 20% of iodide molecules are enters into the thyrocyte cell. Once this iodide enters into the thyrocyte cell, they has to be moved towards the thyroid lumen. This is the thyroid follicle lumen. So this is the thyroid follicle lumen. So this iodide has to be transported towards the thyroid follicle lumen. Once they reach, this is the apical membrane of the thyrocyte cell. This is the apical membrane of thyrocyte cell. Once this iodide reaches towards the apical membrane of the thyrocyte cell, on the apical membrane there is a presence of one enzyme. This is one enzyme that is called as thyroid peroxidase. This enzyme is present over the apical membrane of the thyrocyte cell. So once the iodide comes and goes towards the apical membrane of this thyrocyte cell, this iodide, in the presence of enzyme that is thyroid peroxidase, this iodide get further iodinated. That is, this iodide get converted into the iodine molecule. So the first function of the thyroid peroxidase enzyme is nothing but the first function of thyroid peroxidase enzyme is the iodination of the iodide molecule. So iodide is converted into the iodine. This is the one end of the cycle. Let's go towards the other end of the cycle. Let's go inside the cell. Inside the cell, there is a nucleus at the center. So nucleus consisting of chromatin materials. So let's draw with another pen. So this is the chromatin material inside the nucleus. So thyroid follicle stimulates a particular gene present on the chromatin material. Suppose this is a one particular gene which gets stimulated. This stimulated gene further synthesis mRNA, that is nothing but the messenger RNA. This messenger RNA is a special type of messenger RNA. This special type of messenger RNA further get translated and it is get converted into the one protein. So this is the one particular protein. This is the special type of protein. This is not a normal protein. This is something special type of protein. This protein consisting of sequence of amino acid. This is the one particular sequence of amino acid. Then it contains a large tyrosine molecule. Again the sequence of amino acid, again tyrosine molecule, again sequence, and again tyrosine molecule. So this is not a normal protein. This is something special type of protein in which the tyrosine molecules are incorporated inside this protein. So the number of tyrosine molecules are more inside that protein. So this is a something special type of protein. So this protein, the name of that protein is nothing but thyroglobin. So the name of that protein is nothing but the thyroglobin. Thyroglobin, this is the protein which consisting of number of tyrosine molecule. The tyrosine molecules are more inside. So this protein is further enters into the endoplasmic reticulum. So this protein is further enters into the endoplasmic reticulum. So once this protein enters into the endoplasmic reticulum, it get glycosylated. So this protein once enters into the endoplasmic reticulum, it get glycosylated and get converted into the glycoprotein. So this protein is further get converted into the glycoprotein. So in the endoplasmic reticulum, the glycosylation of this protein happens and it converted into the glycoprotein. So from this endoplasmic reticulum, this protein is further trapped into the vesicles. This protein is further trapped into the vesicle, and then this vesicle is further goes and enters into the Golgi apparatus. So this protein which is enclosed into the vesicle get further enters into the Golgi apparatus, and in the Golgi apparatus the final packing of this protein happens and it further moves towards the apical membrane of the thyrocyte, and finally once it reaches to the apical membrane, this vesicle once reaches to the apical membrane, it get rupture and further there is a release of thyroglobin molecule. So this thyroglobin molecule enters into the lumen of the thyroid follicle. Here there is iodine, here there is a thyroglobin molecule. So I will draw this big thyroglobin molecule here. These are the number of sequence of amino acid. Again there is tyrosine molecule. again there is tyrosine molecule, again number of amino acid, again 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 tyrosine molecule. So number of amino acid, tyrosine molecule, and final tyrosine molecule. So this is the big thyroglobin molecule. The each and every thyrocyte which forms the one thyroid follicle secretes a number of thyroglobin molecule inside the thyroid lumen. So this is a one particular thyroglobin molecule which consisting of more units of the tyrosine molecule. It contains more unit of tyrosine molecule. So the second function of this enzyme, once the thyroglobin enters into the thyroid lumen, the second function of thyroid peroxidase enzyme is nothing but the organification of iodine. This is the iodine, and organification of iodine is nothing but the this iodine comes and goes and finally binds with the tyrosine molecule. Once this iodine goes and binds with the tyrosine molecule, this iodine get organified. So the second function of the thyroid peroxidase enzyme is the organification of iodine molecule. The second function is nothing but the organification of iodine molecule. So this iodine goes and binds to the tyrosine molecule. So the second function of thyroid peroxidase happen and the iodine get organified. Finally iodine get organified. So this is one tyrosine molecule and this is the next tyrosine molecule. So the third function of thyroid peroxidase enzyme is the coupling reaction, that it combines or it binds the two adjacent tyrosine molecule. The third function of thyroid peroxidase enzyme is nothing but the coupling reaction, that it binds, let's draw with another pen, that it binds the two adjacent tyrosine molecule. Once this two adjacent tyrosine molecule get binds, it further converts, this molecule is further convert into the T3. That is, it contains a 3 iodine molecule, that is triiodo, and 2 tyrosine molecule, that is T3. Again the function happens and the two adjacent tyrosine molecule get attached. This is called as T4 molecule. This is called as T1 molecule. Theoretically this is called as T1 molecule. This is called as T2 molecule. So this is the monoiodotyrosine, that is MIT, monoiodotyrosine. This is a diiodotyrosine. This is a T4 and this is a T3. So finally there is a formation of T3, T4, T1, that is monoiodotyrosine, T2, that is diiodotyrosine. Finally there is a formation of T3, T4, T1, T2, that is monoiodotyrosine and diiodotyrosine get happens. So third function of thyroid peroxidase get complete.